Hello, everyone, and welcome to today's webinar, RT-LAMP, Best Practices and Tips to Accelerate Detection of Viral Pathogens, including SARS-CoV-2. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. In addition to submitting questions, we encourage you to participate in today's webinar by answering poll questions dispersed throughout the presentation. When the poll question is displayed, please select your answer by clicking on your screen. Your participation is greatly appreciated. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain your credits. I'd like to now welcome our speakers. Ingrida Vendelė, R&D Manager for Molecular and Synthetic Biology, Thermo Fisher Scientific, and Leann Buhrow, R&D Scientist, Molecular Biology, Thermo Fisher Scientific. Ingrida and Leann, you may now begin your presentation.
Hello, everyone. My name is Ingrida, and I'd like to start by welcoming everyone again to this webinar focused on RT-LAMP. Today, we will start with a brief technical introduction to RT-LAMP as a method, and then share advice on how to set up your workspace when working with this highly sensitive technique. We would also cover LAMP primer design recommendations and share a simple practical example of primer validation process. We will also demonstrate how to generate a synthetic RNA control, which could be used to validate your reverse transcription LAMP results. And finally, we'll finish by sharing practical RT-LAMP solutions for SARS-CoV-2 research and surveillance. LAMP stands for Loop-mediated isothermal amplification. It is a fast, simple, and specific DNA amplification technique under isothermal conditions. DNA polymerase used in LAMP has a strong strand displacement activity, meaning that amplification of DNA can occur at a single temperature because there is no need for a denaturation step to separate DNA strands. When RNA is used as a template, a reverse transcriptase can be incorporated into the reaction to convert RNA into copied DNA. Briefly, LAMP uses 4 to 6 primers, as demonstrated on the right. Amplification is initiated by a strand invasion of an inner primer and subsequent synthesis of an inner sequence. The inner primer also contains a complementary sequence, and following annealing of an outer primer which displaces the first product, the complementary sequence allows the formation of a self-hybridizing loop at the end. And this formed dumbbell structure then serves as a seed for subsequent exponential LAMP amplification. It has multiple sites now for amplification initiation. The end products are very long, which are also connected by single-stranded loop regions into long concatenars, or often also called cauliflower-like structures. The main LAMP advantages are related to simplicity and flexibility. Since it is an isothermal technique, no thermal cycler is required and only a heating block is sufficient. It offers a fast turnaround time from 15 to 30 minutes or even shorter sometimes, and also multiple detection solutions from real-time analysis to endpoint detection. It is a simple workflow with only a few steps, and as mentioned, if RNA is the target, simply incorporating reverse transcriptase is enough. And all these features make LAMP compatible for use in low-resource or in-field settings. When choosing to use LAMP, it's really important to pay attention for a proper primer design in order to control for a false positive signal, and hopefully today we'll provide you good recommendations and insight on how to do that. So now you should see the first poll question on your display, and I'd like to encourage everyone to participate. I'd like to provide some recommendations to how prepare your workspace in order to avoid false positive signals as mentioned before. And when preparing bulk LAMP experiments or simply handling a large number of samples during your assay development, there's always a risk of workspace contamination and resulting false positive signal. So general guidelines for working in the lab apply, such as wearing a clean laboratory coat or gloves while performing experiments, washing your hands prior to working in the lab, and changing gloves frequently during the experiment, especially after cleaning your workspace, such as laminar flow hood, or prior to adding your no template controls or negative control samples. Always follow the recommended cleaning instructions prior and after each experiment as provided in the protocol or a user guide, and just generally follow good laboratory practice guidelines. But when looking at LAMP-related guidelines, I'd like to specifically highlight that using separate workspace areas for RT-LAMP reaction preparation and amplification from RT-LAMP amplicon analysis by endpoint detection when post-reaction tube opening is required is specifically important to avoid a false positive signal. Discard used reagents if you suspect a carryover contamination and replace with fresh ones. Aliquot your reagents into working stock volumes, such as aliquoting primers or your RNA DNA targets or internal controls is good practice. And always use filtered pipette tips, and of course change your pipette tips between your technical replicates. So if you're preparing LAMP reaction to detect a DNA target, we recommend to clean your lab space with in vitrogen DNA ZAP cRDNA degradation solution to remove any potential contamination amplicons from your surroundings, and then clean your workspace with the RNase-free distilled water to remove residual solution, and decontaminate your workspace with a 70% ethanol solution. However, if your target is an RNA sample, then also clean with your in vitrogen RNA ZAP RNase decontamination solution to remove any potential RNases that could degrade your RNA target, and then remove the residual solution with a 70% ethanol solution. However, the most important recommendation is really to not open tubes or wells after LAMP amplification in the reaction setup space to prevent amplicons spreading on the workspaces and contaminating, causing a false positive signal in your new reactions. Also seal your no template control tubes or wells prior to adding a target RNA or DNA. And if contamination is observed, also consider using a different primer system, targeting potentially another genome region. As well, prepare negative controls in a separate area to avoid a false positive signal. And also, multiple technical replicates are always recommended for your samples and for your controls to properly validate your reaction outcome. And here's a simple example of a workspace when multiple reactions are being set up. So here we're looking at a laminar flow hood, which contains cool racks to store your reagents as well as your reaction mix if they're temperature sensitive, and gloves are worn and filter tips are used during the preparation of multiple technical replicates. Contamination can be recognized easily when real-time detection method is selected to either screen multiple primer sets or to observe your reaction kinetics. And in such case, a melt curve profile can be examined, and usually a specific amplification produces a sharp, narrow peak, and here it's highlighted in red, to also demonstrate that your technical replicates overlay nicely each other, showing a specific amplicon. However, non-specific amplification can be identified by observing double peaks or wide shoulders, or even a shift in the melt temperature by more than 1 degree. If endpoint detection is chosen, please have multiple negative controls, as indicated here in the orange colored tubes, and observing a single color change which would indicate a false positive, separating it from the rest of your technical replicates gives a good idea that this is potentially a cross-contaminated reaction mix. Just checking it on an agarose electrophoresis gel, you can observe a blur pattern in the background which indicates a false positive potentially resulting from a carryover contamination in the reaction mix. As mentioned, LAMP primers are crucial for successful LAMP amplification, and here are some guidelines on how to successfully design primers. LAMP employs 4 to 6 primers binding 6 distinct regions of the target DNA, and even though reaction can be set up using 4 primers, so inner and outer primers, addition of the 2 loop primers significantly speeds up amplification. Inner primers are hairpin-forming primers that hybridize to the complementary and reverse complementary target sequences, which forms the dumbbell-like structure and leads to the auto-cycling lamp. The target sequence is also important in successful LAMP assay design, and if faced with high sequence variation from strain to strain and aiming to detect multiple strains, select conserved regions across strains and variants to avoid false negative results, or also avoid conserved genes from closely related organisms to avoid a false positive result. And BLAST can be used to check for non-specific targets. Select homologous regions across strains to detect multiple related strains, and if aiming to detect multiple strains, you can also use multiple primer sets or degenerate primers. Key considerations for successful primer design are very similar to general PCR primer rules, so GC contact needs to be monitored and limited, as well as the melting temperature of the primers is important, and LAMP is usually carried out at 65 degrees, so that should also be the melting temperature with your primer design. It is important that primers do not form secondary structures, especially for the inner primer, which is longer and forms the dumbbell structure. And to prevent the formation of primer dimers, it is also important to ensure that the 3 ends are not complementary. Avoid runs of 3 or more of one base or dinucleotide repeats, and if restriction enzyme sites exist in the target sequence outside the primary regions, they can be used to confirm the amplified product. In such case, you would see a single band on the gel instead of a signature lamp ladder-like pattern. Check distances between target regions as provided here, and also monitor hairpin melting temperature. Here I'd like to present an online tool for LAMP primer design, which is the most popular free tool to design primers since it's cited in the majority of literature. It's called Primer Explorer. It has been developed by LAMP inventors, and the link is provided here on the slide. It has a very descriptive, informative user guide and manual, which really provides a lot of useful information how to design successful primer sets. It has some limitations. It's limited by 2,000 base pair target sequence. It supports only ATCG characters and doesn't handle multi-sequence alignment representation with degenerate nucleotides, which is often a problem when trying to design loop primers, so we suggest not to use degenerated nucleotides. It's limited to a single execution process, and the output is in the HTML format. But despite this, it's an excellent tool. It's used very frequently and cited in literature, which really indicates that the primers work well. And as mentioned, it has a very descriptive user guide for more information. So now I'd like to present a very simplified primer validation process example. And here we're looking at the target of synthetic measles RNA. And primers here are either custom designed using the tool that I just presented or taken from published literature. And the materials for LAMP were in Vitrogen superscript 4 RT-LAMP master mix, in Vitrogen Cyto9 green fluorescent nucleic acid stain for real-time detection to observe reaction kinetics, and of course HPLC purified primers with the concentrations described at the bottom left corner. So at the top, we're looking at primers from publications, and at the bottom, custom designed. And what we are looking at in the LAMP kinetic profile is fast reaction speed. We want to have a fast LAMP outcome. We want to see good reproducibility between technical replicates, so they should overlay each other despite the RNA copy input. And we want to have a wide, long specificity window between specific amplification and non-specific amplification start, which is indicated by green curves. So if looking at publication primers, at the left, we have a speed of just 10 minutes. The technical replicates are very reproducible. Specificity window is about 35 minutes, allowing for a fast and specific reaction. And then on the right, we have non-specific amplification overlaying your specific curves. Reproducibility is not great, and technical replicates are spread out, and we would have difficulties in separating a false positive signal from a specific amplification here. At the bottom, custom design primers are compared, and even though they are custom designed, they do not show equal performance. So on the right, design A, even though it doesn't show non-specific signal, the reproducibility is poor, variability is high, technical replicates are spread out from 10 to 20 minutes, while on the left we have a speed of just 5 minutes, technical replicates overlay each other, and specificity window is the longest, about 40 minutes. So both publication A primer set and custom design B show good performance as we would like to have. And here we selected simply custom design B because of the speed. It's the fastest speed, but both show good performance and could be used. And hopefully this example provides you information on what to look for when examining LAMP kinetic profile. And finally, I'd like to present on how to design a positive control for your RT-LAMP outcome validation. So using positive and negative controls is recommended to validate RT-LAMP reaction outcome. And when considering your negative controls, there are several options. So a no template control, where a reaction mix with water instead of a target can be used. A negative clinical sample, when we know that a target is not present in the sample. A sample with another causative agent. So for instance, if aiming to detect measles, influenza could be used to make sure that your primers do not bind non-specifically to another causative agent, or universal human RNA sample. If human clinical samples are being used, there is a likelihood of background RNAs being present, and this is also a good negative control. And for positive controls, they can be either made in-house or purchased commercially. And if choosing to make your RNA control in-house, there are two possible workflows, and the most frequently used is cloning into expression plasmid to target DNA, and then linearizing, purification, transcription steps can take several days. So today I'd like to present an alternative workflow which only takes a few hours and uses GNR strings DNA fragments. So synthesizing RNA using GNR strings DNA fragments can shorten your workflow significantly. And here we're looking at 3 gene strings that we used to produce our internal RNAs. Double-stranded DNA strings contain T7 RNA polymerase promoter upstream, and also had restriction sites added on both ends, just in case downstream cloning application is planned, but they are optional and absolutely not required. Such gene strings can be used for in vitro transcription reaction without the need for cloning directly into the transcription reaction, which shortens your workflow significantly. Gene strings are supplied highly pure, which again helps to ensure your transcriptional yields are great, which is important, and simplified workflow to generate high quality yields of synthetic RNA fragments within only a few hours is presented. So instead of days, you can have your RNA much quicker. And here are steps. So reaction setup is about 15 minutes only, and RNA synthesis using Thermo Scientific Transcript AT7 high-yield transcription kit is about 2 hours. And then purification using the Invitrogen Mega Clear transcription cleanup kit is about 20 minutes. And on the right, you can see the amount of synthetic RNA that was generated following just these 3 simple steps. At the bottom, we have electropherograms of synthesized RNA fragments. We can see sharp peaks of our target RNA of expected size with 3 different strings that were used in our reactions. And here, positive controls for RT-LAMP are validated. So first, transcribed RNA was quantified via real-time RT-qPCR. Here I'm only presenting gene string C, and you can see the copy number that was calculated from RT-qPCR results. Many copies were generated directly from just 1 microgram of custom DNA gene strings. And real-time RT-LAMP using cyto9 green fluorescent nucleic acid stain demonstrated that RNA transcribed from gene string C demonstrated equal performance to commercial control or purified clinical RNA. And the time to result was just 5.8 minutes when commercial control was just 5.2, and we have 2 good examples of clinical samples with high or low RNA copy number, and the reaction time was very similar. You should now see the second poll question on your display, and your participation is greatly appreciated. I'd like to now pass to my colleague Leann to present RT-LAMP solutions for SARS-CoV-2 research and surveillance.
Thank you, Ingrida, and hello, everyone. My name is Leann, and I'll be presenting the last portion of our webinar. We're focused on RT-LAMP solutions specific for SARS-CoV-2 research and surveillance. SARS-CoV-2's emergence, spread, and health outcomes have impacted all of our lives. Perhaps one of the most important lessons we learned, however, is the need for accurate, sensitive, and available molecular testing solutions for infectious disease. Displayed here is a snapshot of the United States Centers for Disease Control and Prevention's timeline of the appearance and response for SARS-CoV-2. In February 2020, only 3 months after the first pneumonic cases were reported, the U.S. and many other nations identified the immediate need for widely available molecular tests. Today, mass vaccination has been possible in the Americas, Europe, Australia, and many parts of Asia. However, molecular testing remains critical for unvaccinated groups, including within emerging nations, for young children for whom the vaccine is not available, and within vaccine-hesitant populations. Furthermore, with the evolution of viral variants, molecular testing provides us with epidemiological data on the incidence, distribution, and impact of vaccination on SARS-CoV-2-derived pneumonia. Let's discuss the SARS-CoV-2 virus in greater detail. Severe acute respiratory syndrome coronavirus 2, or SARS-CoV-2, is a positive-sense single-stranded RNA virus. This virus attaches to host cells using its eponymous crown-like spike proteins. In the diagram on the left, the viral RNA is shown in red and these spike proteins are shown in orange. SARS-CoV-2 molecular tests detect either this RNA or spike protein. As SARS-CoV-2 infections continue to spread, novel variants have evolved. These variants of interest have small RNA changes in the S gene, resulting in amino acid changes in the spike protein. These changes may either help the viral variants exhibit better host cell attachment, increase transmission or viral load, or improve the ability of the virus to evade the host antibodies. With so much research and development on SARS-CoV-2, it can be a challenge to understand and relate all the information that's been given to us. Here, we're outlining 2 of the most common attributes. First, SARS-CoV-2 test types, as well as the required sample types. SARS-CoV-2 is principally a lower respiratory disease. However, viruses may be detected in minimally invasive sample types like saliva, nasopharyngeal swabs, and nasal swabs. Additionally, the human immune response to the virus may be observed using a blood sample type. Shown on the right, there are three common types of SARS-CoV-2 testing: nucleic acid-based, antigen-based, and antibody test. Nucleic acid-based tests detect SARS-CoV-2 RNA. This is colored in red. These tests may be called PCR or qPCR tests and are very sensitive and specific. Most nucleic acid testing requires trained scientists and a centralized laboratory to determine if the virus is present. Therefore, these tests often take hours to days to perform and may be expensive. Alternatively, antigen-based tests or rapid tests detect SARS-CoV-2 proteins. These tests may be performed at the point of care without extensive training or specialized instrumentation. These type of tests can tell a patient whether or not SARS-CoV-2 is present at very high levels only. Therefore, this fast, inexpensive test will identify those patients that are likely contagious. Finally, antibody tests detect the human immune response to SARS-CoV-2 exposure. These tests can inform a patient on whether they recently had or have an infection, and the results are typically provided with a quick turnaround time and little financial investment. In addition to the common SARS-CoV-2 tests we just discussed, RT-LAMP may be used for SARS research and surveillance. On the left, we're depicting SARS-CoV-2 viral load variation, where a person on the extreme left, in yellow, only has a low viral load. This low viral load may be detected with qPCR tests only because these tests have high sensitivity. On the right, the person in red has an extremely high viral load. This high viral load may be detected by any test, including the antigen tests, which have the lowest sensitivity. RT-LAMP-based tests for SARS-CoV-2 are uniquely positioned as they're nearly as sensitive as the qPCR tests and are nearly as fast as the antigen tests. The application of RT-LAMP is so well suited for SARS-CoV-2 that the landscape of this approach has drastically shifted over the last 2 years. As seen on the right of the slide, in 2019, only about 2% of RT-LAMP publications were focused on SARS-CoV-2. However, in 2020, this metric drastically shifted, with approximately 60% of all LAMP publications focused on SARS-CoV-2 research available. Thermo Fisher Scientific is invested in providing RT-LAMP-based solutions for infectious disease detection, including SARS-CoV-2 research and surveillance. In the last year, Ingrida's team and my team have been focused on two specific offerings. The first is the Invitrogen Superscript 4 RT-LAMP Master Mix. This is an optimized enzymatic master mix resulting in outstanding RT-LAMP performance. A master mix provides the ultimate flexibility, allowing the user to select their pathogen of interest, their target, and their readout method. The second solution is the Invitrogen Colorimetric ReadiLAMP Kit. This is an off-the-shelf complete kit for SARS-CoV-2 detection in saliva or NP or nasal swabs. 2 reaction controls and 2 workflows optimized for sample types are included. As with the Superscript 4 RT-LAMP Master Mix, the Colorimetric ReadiLAMP is intended for RUO-only applications with minimal cost or instruments investments required. Okay, now I'm going to focus on the SARS-CoV-2 research using just the complete kit. The Colorimetric ReadiLAMP Kit provides 2 workflows, one for RNA samples and one for crude samples. In the case of RNA sample type, the user collects saliva, nasal or nasopharyngeal swabs. SARS-CoV-2 RNA is isolated from these samples, and then finally, the isolated RNA is used in the Colorimetric ReadiLAMP Kit as sample input. Here, the user assembles an RT-LAMP reaction, incubates at 65 degrees for 30 minutes, and then interprets whether the saliva or swab sample type contains SARS-CoV-2 based on colorimetric change. Samples lacking SARS-CoV-2 will remain purple in color, while samples containing SARS-CoV-2 will turn from purple to blue color. For the crude sample types, this exact protocol is used with the RNA isolation step omitted. SARS-CoV-2 from these samples is inactivated and lysed and directly added to the colorimetric ReadiLAMP assay. Here, we added a poll question. We're wondering what type of sample is best for you in your research. Let's look at some of the SARS-CoV-2 detection data with a colorimetric ReadiLAMP kit. For RNA samples, these are shown on the left, the complete workflow, including RNA isolation, was used. 96 saliva and 96 MP samples were processed, where half of each sample type contained the SARS-CoV-2 virus. The RT-LAMP reactions containing 100 copies of SARS-CoV-2 from both samples all turned from the initial purple color to the positive blue color, demonstrating positive overall reaction, while all reactions lacking SARS-CoV-2 remained purple in color. For the crude sample types that are shown on the right, the workflow omitting RNA isolation was implemented. In this case, all RT-LAMP reactions containing 250 copies of SARS-CoV-2 turned blue, while reactions lacking SARS-CoV-2 remained the initial purple color. Overall, with both sample types, superior performance in terms of RT-LAMP accuracy and specificity are recorded. One critical question about the colorimetric ReadiLAMP kit is, does the RT-LAMP reaction detect SARS-CoV-2 variants? SARS-CoV-2 variants have a key RNA change in the S gene, but don't have concomitant changes in other regions of the genome. The colorimetric ReadiLAMP primers don't target the S gene and therefore should capture all viral variants of concern. In fact, when we investigated more than 633,000 deposited SARS-CoV-2 genomes, more than 98% are perfectly complementary to the kit primers. Furthermore, we purchased 15 available SARS-CoV-2 variants. For all 15, the colorimetric ReadiLAMP kit detected the presence of the virus at 50 copies per RT-LAMP reaction, supporting that this kit does detect SARS-CoV-2 variants. Finally, we demonstrate that the colorimetric ReadiLAMP kit is specific for SARS-CoV-2 and does not suffer from false positives in the presence of other respiratory pathogens. We tested isolated nucleic acid from 12 saliva donors containing either flu A, a pooled respiratory panel, SARS-CoV-2, or no pathogen. The respiratory panels tested herein are pooled aliquots of 8 to 10 causative agents of respiratory disease. As you can see, only RT-LAMP reactions that contained 100 copies of SARS-CoV-2 turned from purple to blue and thus are called positive reactions, while all other RT-LAMP reactions remained purple in color and are negative. This data fully supports the specificity of the colorimetric ReadiLAMP kit for detecting SARS-CoV-2 and differentiating between different types of respiratory disease. We hope you now have a better understanding of how RT-LAMP may accelerate detection of viral pathogens, including SARS-CoV-2. Today, we provided a theoretical introduction to LAMP technology and practical information on the RT-LAMP workspaces, primer design and validation, and synthesizing RNA controls. Furthermore, we described the Thermo Fisher Scientific RT-LAMP solutions for research and surveillance. Here, we have an additional poll question focused on RT-LAMP applications. We'd appreciate your feedback. Thank you so much for your attention today. Ingrida and I would love to address any additional questions and look forward to that in the Q&A session at the end. We have one additional poll about learning more about RT-LAMP solutions.
Thank you, Ingrida and Leann, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, can you use unpurified or minimally processed samples with the RT-LAMP master mix?
Hi, thanks for the question. So Invitrogen Superscript 4 RT-LAMP master mix was developed using purified RNA samples. However, the BST DNA polymerase, which is included in this product, is described in literature to demonstrate improved processivity and good inhibitor tolerance. So we highly recommend trying and optimizing techniques for direct LAMP described in literature with this product, and hopefully we'll have some recommendations soon as well.
Thank you. Our next question is, what method do you use to quantitatively record the color change with the Colorimetric ReadiLAMP Kit?
So the Colorimetric ReadiLAMP Kit can be read with a naked eye, just monitoring that purple to blue color transition. However, we understand that some people may want a more quantitative measurement. So to address this need, we have application notes available on our website for using the Colorimetric ReadiLAMP Kit with a spectrophotometer or a plate reader. So this should help users who want to not only use a quantitative measurement using the instrument, but also anyone who's interested in high throughput applications. So the Colorimetric ReadiLAMP Kit application notes can be found at www.thermofisher.com/ReadiLAMP.
Thank you. Next question. When analyzing crude samples with the Colorimetric ReadiLAMP Kit, how is the SARS-CoV-2 virus inactivated in the samples?
Right, that's a great question. We also want to note that being very careful with how you handle the SARS-CoV-2 virus is paramount for any research. So here, what we're suggesting is that the user inactivate the SARS-CoV-2 virus and lyse it using a combination of a 95-degree heat step in the presence of a reducing agent. The Colorimetric ReadiLAMP user guide has this protocol incorporated. It only adds 5 minutes to your overall workflow, and it can help you and your lab be safer and be less exposed to the virus.
Great, thank you. Next question. What RNA isolation kits are compatible with RT-LAMP solutions from Thermo Fisher?
That's another great question. So for the Colorimetric ReadiLAMP kit specifically, we've tested a variety of commercially available RNA isolation kits, and they all work great. We've done the most work validating the magnetic bead-based MagMax kits, as well as the column-based PureLink kits, and those are really great for superior overall RNA isolation.
Thank you. Next question. How can I distinguish specific amplification from non-specific?
Thanks, that's a very important question. So as mentioned, LAMP is highly sensitive and produces amplicons in a form of concateners or cauliflower-like structures that vary in size but contain repeats of the same DNA target sequence. So such amplicons usually have a unique melting temperature, which can be used to confirm amplification of a specific target. And when using real-time detection option, a melt curve analysis can demonstrate the maturation profile and help separate specific amplicons from non-specific. So specific amplicons should demonstrate a single sharp dissociation peak, and characteristics that indicate non-specific amplicons are wide shoulders, multiple peaks, a shift in melting temperature. When compared to specific amplicons, that could be 1 degree or more. I hope that helps.
Thank you. Looks like we have time for one more question. What downstream applications are compatible with RT-LAMP amplicons post-reaction?
Okay, that's another great question. So LAMP produces amplicons that vary in length, and are composed of numerous repeats of the short sequence, as mentioned before. So they form these long cauliflower-like structures, and such products are not typically suitable for downstream applications. So if the restriction enzyme sites are added on the target sequence, except the primary regions, then they can be used to confirm unique amplicons via restriction and gel electrophoresis.
Well, thank you, Ingrida and Leann, for your presentation. Do you have any final comments for our audience?
Just want to thank everyone again for joining in, and hopefully this was useful and you'll be tempted to try RT-LAMP soon.
Yes, I'd just like to repeat Ingrida's thank you to everyone at Thermo Fisher, especially our team members, for contributing to all our RT-LAMP solutions. And we really look forward to working on this more, especially with new applications.
Well, thank you again, Ingrida and Leann, for your time today. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.